Thank you very much for the in-depth introduction. And first of all, I would like to thank the organizer for inviting me. So it's really a pleasure to be here. It's my first Synapsa meeting, and I'm enjoying it a lot. So it's like, thank you very much. And first of all the part of my lab, as Ivana said, is doing ILS research. But I was asked in particular by Boris last year that my talk has to be more general about neuroinflammation. So this is how I will focus this talk. So my talk will be divided in, this doesn't work. Pointer. Where do I have to point? I can switch here, but it is. Yeah, so it doesn't work either. Yeah, but I want to. Okay. Okay, so my talk will be separated in a couple of chapters. So first I will discuss the innate immune response in general, and I will brush up about microglial cells in the brain and the function. Afterwards, I will discuss the regulatory mechanisms and some of the very unique molecular signature that we discovered in acutely and chronically activated microglia using our novel model systems. At the end, I will talk about the recent developments of a new therapeutic strategy we have having in the lab. And at the end, I will just show to you a couple of slides with the most recent unpublished data that we have using different models. So our interest in the lab for the past decades was really to better understand how immune cells interact with the brain in health and the disease state. And our particular interest was toward innate immune response and innate immune cells. So innate immune response is actually the first line of our defense. It's activated when we are attacked with bacteria, with the viruses. It is in response to tissue injury and neurodegeneration. And normally it's there to protect us. But once that is inappropriately scaled or prolonged, it may cause the disease. When we looked into a brain, the primary cells that are innate immune cells are called microglia cells. And they play a really instrumental and critical role in initiating and regulating this response. So why we were so interested in microglia? Because these are the cells that are really one that mediate innate immune response in the brain. They were first described at the end of the 19th century by Pio del Ortega. And if you can, where is the pointer here? Okay, I found it. So first described by Pio del Ortega at the end of the century, you can see here about the silver staining that they're extremely ramified cells that are present in the brain. They're called microglia because they were smallest of the glial cells. And what is interesting is this morphological transformation of these cells. So once they are activated, the first thing that was noticed in these cells, they changed the morphology from extremely ramified cells to the amoeboid cells that are really getting into very powerful phagocytes. So, this you will not see well if you can dim the lights. So because we have quite a... So the first description is like you can appreciate here, not very much, but the thing is that morphological description of microglial cells haven't changed from the end of the 19th century to now. So this you can see on this slide is the images of my activated cortical microglia using the confocal microscopy. So what is interesting is that I wouldn't be here talking about these cells if they were not for the phenomenon called microglial activation. And what is interesting here that microglial activation is a hallmark of any brain conditions, starting with brain injuries, chronic neurological disorders, and even infections. So what can activate microglia? Many pathogens, only starting with the neural dysfunction. The stress neurons can activate microglial cells. Protein aggregates, which is really in processes of aging, which is really important in the field of neurodegeneration. And there are some epigenetic risk factors that may include activation of microglial cells. So once activated, microglial cells can develop several of the different phenotypes, functional phenotypes, with the two extreme going to the extremely inflammatory neurotoxic cells that will kill neurons in the brain, or the protective cells that will help them. So this was kind of a initial kind of a pluralized and simplified version of the microglia, but now they're dividing them in the good and the bad cells. So M2 nice versus very nasty phenotype. But the thing is that the whole field of neuroinflammation and the microglial studies on microglial cells evolved, and now we look at the cells as more complex, functionally more complex cells, and that can acquire many different functions and many different coexistent phenotypes at a certain stage. What we don't know now is to what extent these different phenotypes are reversible or not reversible. So this is something they're really looking for into it. And for a look into new nomenclature and how very dynamic this field is, I will invite you really to consult. This is the last review on the new nomenclature and the function of microglial cells that went out by the end of this year in Neuron, and there's about 50, 60 authors that all have a little contribution of the cells. So it's a very useful manual how to better understand microglial cells. So one of the things that we did in the lab for over a decade now, we asked several questions, and by asking several questions, we also developed different model systems. So one of the first questions, we said it would be really nice to visualize microglial cells from the living animals. And how can we do it? So for that, we developed the reporter mouse that's called TLR2 Luc-GFAP reporter mouse. So we choose the TLR2 receptor as a reporter gene, but why? Because TLR2 receptors, TLR2 belongs to the large family of TLRs. These are the receptors upregulated during innate immune response. And once activated, they will signal through adapter molecule MYT88 and activate canonical NF-kappa signaling. So what is very convenient in this sense, normally these receptors are silent, but once our brain and microglial cells are attacked or activated, it will get strongly upregulated, and we'll see what would be the way to visualize this response. So we generated TLR2 Luc-GFP reporter mouse. So the answer was we turned our mice into fireflies. So what is the principle here? So to visualize innate immune response in microglia, we co-express luciferase and GFP as reporter genes under transcriptional control of a murine TLR2 promoter. So every time in this mouse, when you have activation of innate immune response in microglia, you will have this bioluminescent signal, and you can use GFP to co-localize the signal within the microglial cells. So how does it work, really? Bioluminescence is a chemical reaction. So we expressed our luciferase is expressed in microglial cells, but you have to add luciferin, and then during this reaction, it's a double oxygenation. You have the end result is an inert product, oxyluciferin, and emission of the light. So basically, if you have a bioluminescent source in the brain, you will basically pick up the signal at the surface of the skull, and then by optics, we use the CCD camera, high-resolution CCD camera, to basically record the signal. And this is how it looks in vivo, actually. So this is the mouse who have a stroke, and we have basically, we can see this signal from the living animal. Importance of it is that we can follow the same animal over time, and we can follow the activation of the innate immune response in microglial cells in the living mouse. So, for example, one of the first studies we did in the model of ischemic injury, so we recorded, these are the recordings of the same mouse that we follow for 3 months, and we follow this mouse longer as well, up to 6 to 8 months. So we can see the progression. This is before ischemic injury, and we can see the progression of the signal after the injury, up to 3 months. And what is very interesting also, we can quantify it, and we see very interesting patterns. So as expected, we recorded acute induction of microglial activation after ischemic injury. But then what was rather interesting and puzzling is that we follow it over 3 months, but even 6 and 8. In imaging, this signal, once you have ischemic injury, this signal never goes back to normal. So it seemed that there is a kind of ongoing chronic inflammatory activation of innate immune response. And at this point, we don't know whether this is the Whether this is the resolution, whether this signal is involved in the resolution of the injury, or there is a really risk for further pathologies. So we can also use the spectral imaging and follow the signal, evolution of the signal in the brain of living animals. So this is very nice. We can follow the signal, we can see functional output of innate immune response, but we don't know really what is the function of these cells at the end. So for that, we were actually one of the first to show that microglial cells have very useful and neuroprotective function. So in the mouse model where we can ablate microglial cells from the ischemic brains, we actually observe that once we ablate microglia from the brain, we have a larger infarction and we have increased activity of caspase-3. Cleaved caspase-3 activated caspase-3. And what was very interesting when we looked at the cells who were caspase-3 positive, we see when we remove the adequate microglia response after stroke, we have an increased neuronal apoptosis and larger infarction, suggesting that microglial cells carry protective function. And in the sense, when we looked into the cells and see what is really going on, we see that most of the cells that are proliferating acutely after stroke, they were positive for IGF-1. So basically what we were doing when we were ablating cells, we were ablating also the cells that were secreting anti-apoptotic and pro-regenerative growth factor IGF-1. The similar stories was published a bit later by the group of Adam Danish, so he used a different model system. I was very happy to see that they were able to repeat our study. So basically they used the CSF1 antagonist, they ablate chemically microglial cells, and they also observed that once you don't have properly functioning microglia after ischemic injury, you would get larger infarctions and you will have a problem with neuronal networks and function. Altogether suggesting that microglial cells are there really needed to have a proper and innate immune response are there to help to injury. So the other function is that we wanted to go a little bit more in detail. So from visualizing activation of the cells and seeing what cells are doing, we wanted to go to understand more what is going on within the cell type, and we wanted to go and actually analyze molecular profiles of these cells at the different time points after injury or disease. So to do that, we developed a mouse model who would express larger ribosomal subunit on the CD11B, myeloid CD11B promoter, and they were fused with EGFP and FLAG, so we can basically immunoprecipitate a ribosome from activated microglia anytime and see what is their molecular profile from activated microglial cells. So we modified classical TRAP protocol, translational profiling protocol, because once we isolated, we pulled down the ribosomes from the activated microglial cells, we perform parallel RNA and protein analysis. So basically, we use a messenger RNA as an input and protein peptide as an output. So at this time, we will exactly know, so what is coming in, what is dynamic translational state of the microglial cells, and what are the differences between transcriptome and proteome within the same cell population. So as a first step, we use the classical LPS challenge. So this is because we know this model. So we know once you induce systemically LPS doesn't cross the blood-brain barrier. We don't have a contribution as well of the CD11B positive monocytes from the periphery. But there is a huge wave of activation and peak of transcriptional activity 24 hours after systemic LPS. So what we found is that when we compare the transcriptome and proteome of these microglial cells, is that the huge cluster of LPS, so-called acute immune genes, called LPS genes, were there. So we were happy to have a positive outback. But what we've seen is that these genes were not translated. Although they were in a position, they were attached to polyribosomes, they are in the time they are basically actively translated, but something arrested them. So these genes, for example, as SIR3, the human equivalent is SIR2, it was overexpressed at RNA level close to 30 times, but it was not detected as a peptide. So this is the confirmation they were not regulated. We did a cell-specific Western blot and elution Western blot analysis. So when we compare the proteome and the transcriptome of these cells, the activated microglia cells look like schizophrenic cells. So at the RNA level, everything was highly upregulated, and they have a really profile of innate immune cells. All the immune networks were highly activated, and the function of the cell was really immune cell. At the protein level, most of the proteins were downregulated, as observed here by green, and most of the function that were upregulated at homeostatic and regulation of the cytoskeleton. So no immune function was basically upregulated at 24 hours after LPS related to really immune function. And this was most prominent when we looked into NF-kappa B network, because all of the members of the network were highly upregulated at RNA level, but the proteins were not regulated, suggesting that there is a dichotomy between RNA profiles and protein profiles in activated microglial cells. So the question was, what is really going on? So first we thought, okay, so this is what we're going to do. It was against the dogma, because what was really thought is once the RNAs, mRNAs are aligned on the ribosomes, they're actively translating. So this is not that we are selecting general RNAs from the cell. No, these are the ones that are supposed to be translated. So we looked into potential mechanism, and this slide probably took us a year or something. So when, and there was no question of mRNA stability, we checked that as well. So it's what we looked into it, we focused our study on SIR3. So this is the acute immune gene that was upregulated 30-fold at RNA level, not regulated as a peptide or protein. And we took because for most of so this is not the translational regulation. For most of the post-translational regulation, the mechanism would target 3' UTR region of RNAs. So we looked into 3' of the SIR gene, and we checked for the potential regulatory element. We had the luciferase assay to capture it. So we did several of the deletion mutants, and what we see, we were very lucky because the 3' UTR was very small, so we can dissect it really in the small sections. So what we see that there is a kind of a dosage response, suggesting that all of the 3' UTR parts were actually containing regulatory element. So we did plenty of bioinformatics studies and rest. So 1 year later, we've seen a certain pattern, and what we observed is that all regulatory elements, the only protein that we could detect was the RNA binding protein called SRSF3, and it was basically highly the binding sites were highly enriched in all parts of 3' UTR. So we were surprised because we didn't know anything about this protein. So we looked a little bit into literature, and most of the finding that we've seen was linked to a cancer biology. It's a proto-oncogene, is a RNA binding protein involved in the viral responses. So was known basically about the role of its role in immune system and the role of microglia. So this is something that we really started from the scratch, and we looked into. So it's really a protein that is heavily involved in RNA metabolism, and we actually, our next step was to really examine how it is regulated in the microglial cells and how it affects the function of these cells. So what we first looked was, this is in vitro work. So first we looked whether in neuroinflammatory condition we can upregulate this protein. So basically these are just the BB2 cells, the mouse microglia. We looked actually that the total proteins is not changing, but in neuroinflammatory condition we highly upregulate the active form, it's a phospho-SRSF3, increasing the level of phosphorylated protein, and total protein together. So we asked what would happen if we silence the SRSF3. So we silence it using siRNA targeting SRSF3. So what we did is basically when we measured the level of protein by Western blot, we see de novo synthesis of siRNA3. So once we do the knockdown of the protein, we have de novo synthesis of the targeted gene. So this is quantified here, and this is we looked into a luciferase assay, so we restored the luciferase activity in the assay, and this was the quantification of the knockdown. So we know already, we detected that there are several binding sites for SRSF3 in the 3' UTR region for SA3, and we looked into other genes from the cluster LCN2, CCL3F5, and all of the 3' UTRs were heavily enriched in the 3' UTRs of these genes, and we looked in the cell. So when we knocked down when we silenced SRSF3, we have de novo synthesis of this protein as detected in a cell-specific Western blot. So this was in vitro, and we took now here advantage of our TLR2 model to see what is really happening. So basically, what we did is we introduce the siRNA intranasally. We wait for the 48 hours. We confirm the knockdown, and then we did the LPS induction of the innate immune response. And what we observed is that the mice that receive siRNA compared to the scrambled sequence have a higher activation pattern of the microglia. So that would have been as expected. So we have a higher microglial activity. And when we pulled down the microglia from the brain and did the Western blot analysis, we also observed that basically there were the de novo synthesis of the protein in this. So we can basically, and we also went to the several sections of the brain, and we confirm basically the neat thing is when you do the intranasal injection of RNA and other materials, there is a 95% of the probability that all this material would end up in microglia, because they normally phagocyte any of the RNAs, double-strand RNAs, anything that would resemble to either viruses or the other material. So we observed, basically, we confirmed that the 95% of the siRNA as a SIGLO here as a marker was basically with the successful silencing in the CD11B and EBA1 positive cells. So the other thing, what was really interesting is, okay, so we can downregulate this RNA binding protein that at this point we don't know much about the function. So what is the mechanism apart from it? So we looked into the clusters. I'm really apologizing because the resolution is not great. But what we did some of the analysis of 3' UTRs, because what is important to mention here is that SRSF3, when we looked into RNA translation, it was really, the arrest was really specific for the cluster of highly upregulated innate immune genes. Other RNAs were normally translated and detected at the peptide level. As expected. So this is something really that was specific for the innate immune response and innate immune genes that was not related to other RNAs that we observed. So we looked into 3' UTRs of this cluster of the genes that we had, and we noticed that all of them had a significantly higher number of binding sites for SRSF3 as compared to all other genes. And the other thing is we did kind of the three-dimensional analysis, so I don't have a data. But the 3' UTRs of the regulated genes are really much shorter than any other 3' UTRs of non-regulated genes. So they are shorter, and they have a high density of SRSF3 binding site, and it's when we looked, I didn't put all the data. When you look within the first 200 base pair, it's extremely dense. So this is basically what happened, and that's why most of the genes were really immune genes are kind of predisposed to be regulated by this RNA binding protein. And we did also some of the iClip experiment, and we confirmed that there is indeed physical interaction within the 3' UTR of the selected genes and the SRSF3. So we pulled it down here. So everything is nice. So far this is our working model. So we uncovered that there is additional layer of control in the regulation of innate immune response that is based on ribosomes, that the highly upregulated innate immune genes in acute response are not necessarily translated, and that creates certain divergence between RNA networks and protein networks, which can confuse the function, because function at the end will be ultimately regulated by proteins, not by RNAs. And what we also found, we identify novel role of SRSF3 as a master regulator of innate immune response in microglia and regulator of innate immune gene translation in this context. So what we also found that there is a kind of feedback. So when we knock down and silence endogenous protein, we can really regulate proton of microglia proton, because we can induce de novo synthesis of the proteins of the selected genes that are arrested in translation. So this is our schematic, and we found. So what the next question is whether this mechanism can be implicated in any other disease. So this is control acute activation of microglia cells in vivo, but what is happening in the disease? So I will not go into too much of the details, because we listened a lot about ILS and pathogenesis of ILS here, but I will basically focus on this part, because many evidence suggests of the involvement of non-neuronal cells in ILS, and there are several previous articles. So non-neuronal cells, in particular microglial cells, activation is a pathological hallmark of ILS. Actually, this was one of the first microscopic findings in the section of mouse model as well as in the patients. What has been known so far, that microglial activation really parallels disease progression, and the groups of Stan Appel and Don Cleveland really show that once you replace the microglial cells, sick microglial cells with the healthy cells or wild-type cells, you can significantly modify disease and rescue large part of the phenotype that you can see in this in the mouse model. So another also compelling evidence is that most of the mutation that are of the disease-causing genes in ILS has been associated with the changes of different immune functions. So we know that in CNIO, for example, there is a kind of a pro-inflammatory priming in microglial cells, and there is an impaired regulation of autophagy and lysosomal pathways. Optineurin as well, as Ivana knows, will target NF-kappa B pathway, and the function is changed in the disease. TBK1 also, TDP43 and TBK1 as well, they can impair autophagy, and they change the level of inflammatory signaling. So all of the disease-causing genes, and most of that are directly or indirectly associated with the changes in innate immune system and the function of microglial cells. So there is a good solid base to go forward and to see whether we can detect something on molecular level. So what is the question? So this is the question that we had in the field for a long time. So what is happening with the microglia in ALS? So the current view is that these cells are protective early in disease. They have a protective phenotype, but what is happening later? The question is what is happening at this stage. When they're overly activated, whether we have a loss of normal function or whether we have a gain of toxic function. So these are the two things that are kind of a debate, we have a debate in the field. And what is really important is that once microglia cells switch from this state to this state, they are resistant to any conventional immune modulatory treatments. So what you can do when you treat the cells in this stage, so for example, the SOD1 mice after the onset of disease, what would happen if you treat them with anti-inflammatory treatment, you will aggravate disease. So this is something that has been shown also in the clinical trial. So the question is, we went back to our model, the great model that just show us what is happening with the innate immune cells, and we said this is great. So we can use this model, cross it with the SOD1 model, and look what, and follow the microglial activation over disease. But this was the double transgenic mouse. So this is the wild-type mouse. This is the SOD1 mouse model at the time of the onset. This is the end stage of disease. No single photon is detected, TLR2, reflecting functional innate immune signaling. And we know that the microglial cells at this stage look like here. So they are excessively activated, but they not upregulate functional innate immune signal. So we ask what is going on at this level. So we went to the kind of induction of a functional immunity model because we couldn't record anything. So this is we induced the LPS response in the wild-type mice and age-match littermates of the SOD1 mouse. And what we observe is that there is a markedly diminished response to innate immune challenge in this mouse, and this is in the brain as well in the spinal cord, suggesting indeed that there is a problem with functional innate immune signaling in these animals. So, and this is also what is really nice review what we put together with Ivana is that what is happening, we think, is that once we get to the developed disease, the symptomatic disease in mouse as well as in humans, we have excessive inflammation. But there is another level of inflammation that is coming with the development of disease, and this is inefficient immune response. And that's what we are basically seeing because these microglial cells do not respond well to any of the innate immune challenge. So the question is, what is the consequence? What could be the consequence of dysfunctional immunity, and how does it contribute to disease progression? So some of the responses was very useful. The couple of papers came recently, one from Ido Amit and Michal Schwarz group. This is the RNA, very nice paper showing the RNA-seq data, cell-specific RNA-seq data. And what was really interesting is that both in Alzheimer's disease and aging in ALS, the cells would express the cluster, upregulate the cluster of the genes that are called kind of disease-associated genes, such as cystatin, LIRB4, TREM2, CLEC7, so CD11-3, so-called kind of a disease-associated genes. So, but they were not specific to ALS because they were detected in Alzheimer's disease models, in ALS as well as in aging, and similar analysis came from Leonard Petruccelli and John Fryer's group because they also sequenced the microglia from H, APP, and tau pathology mice. So they discovered the same signature. So CLEC7, cystatin, CD11-3, and other genes that were basically upregulated at RNA level at different stages of disease, basically at the disease time, but in the different diseases, including aging. Suggesting that there is a common RNA profile that is called disease-associated profile, but it's not there only in disease, it's also associated with aging. So we say, okay, we'll test our system and see what we can dig out on this. So what I'm going to show, this is a part of the unpublished recent data that we generated in the lab. So what we did, we crossed our CD11B ribotag mouse with the SOD1 mouse, and here I want to emphasize one thing. So we intentionally picked up the SOD1 mouse, although it is only 3% of the ALS-caused disease. This is the mouse that developed the only fully blown activated microglia profile. And if you want to study innate immune response in the animal model, this is the only ALS model that really has it. It's not we have all other models in the lab, but this is the one that only replicates what is happening in a human disease in terms of reaction of microglial cells to the disease, because it has a massive motoneuronal death at a certain stage, the same thing what is happening in the humans. So this is why we pick up this mouse. And we did the same experiment. Basically, we pulled down the ribosome from the lumbar spinal cord, microdissected it, pulled down the ribosome, and we compared the RNA and the peptides at the 3 different stages of disease. So pre-symptomatically, there is nothing much going on. But at the symptomatic stages of disease, we pick up more or less the very similar gene. We have a very similar DAMG profile. So we have LIRB4, we have CD48 here. This is moderate, but here we have a CLEX7, we have a cystatin, LIRB4. So these are all TREM2, CD11B. So these are all the genes that are commonly upregulating in neurodegeneration and in aging. So this is an RNA level. So what was interesting here is that none of the genes was actually regulated as a peptide at protein level. So we again, when we looked into comparison of immune networks at RNA level, at the protein level, we see the great disconnect as well. So we have extremely enriched inflammatory profile at RNA level, but very few of these genes are actually regulated and expected and found at the protein level. So again, we are facing the same question. So what is happening with the innate immune system in the chronically activated microglia? So our usual suspects of, well, let's check what is happening with SRSF3. So we ran the Western blots, and what we see from the lumbar spinal cord, we see that indeed, starting at symptomatic stage, there is a significant increase in the expression of SRSF3, phospho-SRSF3, and this is increasing when disease parallels with the progression of disease. And what is really interesting, this pathway seems to be not activated pre-symptomatically. So it starts really at the time of the paralysis. So this is directly correlated with the disease. And this we have a kind of a total. We are also working on the ratio of phospho and total RNA, so SRSF3, and we have an increase. And when we looked into cellular expression pattern, we see that the expression of activated forms of phospho-SRSF3 were mainly restricted to CD11, EBA1 positive cells, and were not detected in other cell types. So this is the process that was really kind of specific for microglial cells. So this is in the spinal cord of SOD1 mice. This is in a rat model as well. So in the 2 different species, we obtained the similar results. And this is the human data. So we have a couple of slides here, but here you don't see well. So this is the SRSF3 in the sporadic cases. Expression is restricted to microglia EBA1 like positive cells. And when we looked into CSF, we see significant increase by ELISA in the SRSF3 levels in the sporadic patients. So suggesting that this process seems to be initiated across the species, so in the mouse, in the rat, and the human disease. Our question is, so what is the functional consequence of this and whether we can target it? So to do so, we generated kind of oligomorpholinus because we want to silence the SRSF3 in immune cells, and we wanted to see what's happening. So we target here the part of the 5' UTR as a start codon. As a consequence, the advantage of morpholinus is that they will not go into nucleus. As a concept, they will stay in the cytoplasm. They will bind to the start codon, and basically they will block the translation of the targeted RNA. At the end, you will induce the knockdown of SRSF3. So what we also use, we did a kind of a mini clinical assay in the mouse. So basically, all of our mice were phenotyped in a sense. So as you noticed before, this is the process that is not initiated before the clinical onset of the disease. It's mice. So all of these mice already have a loss of extension reflex and the partial initial paralysis. So the treatment is initiated at symptomatic stage of the disease. To follow the protocol, what we did the intrathecal injection of altogether 10 micrograms of the antisense morpholinus targeting SRSF3. We did a knockdown here of the Western blot. We preferentially ablate phosphoform, but here is the ELISA and ELISA as well. So we obtained significant downregulation of SRSF3. as compared to scramble, but what we've seen is the restoration of the normal ratio between the phosphate and non-phosphate in the control and in the treated mice. So what was there at the biochemical level? We were interested whether by reducing the SRSF3 level, we can induce de novo synthesis of the protein, of the genes that were arrested in translation. So as our output, we use the TLR2 because we know from our imaging study that TLR2 imaging, there is no functional innate immune response in these guys. So we looked to CD11-3, LIR-B4, CLAC7, cystatin, and we see that all of these genes were now synthesized and detected and upregulated at protein level as well. So, and we use the TLR2 as output, so we have an increase in TLR2 expression of TLR2 receptors, suggesting the reinforced innate immune response. So, well, how does it have any therapeutic potential? What we did, we looked into survival and into scoring system and body weight. So what we looked is that we started treatment very, very late. At the 140 days, these mice live about 160. So this is, and average onset in our colonies around 110, 120 days. So we started very late. These mice are all symptomatic. They have hind limbs initial atrophy and paralysis. So in spite of this, we obtained significant, it is one shot only proof of concept antisense approach. So we had the significant increase in survival. We prevented the weight loss and the progression of the symptoms with our score. And what we looked in the spinal cord, we had the increased expression of CHAD-positive neurons, suggesting neuroprotection as well as the end results. And when we looked into microglia itself, so we see that in the mouse that were treated by morpholino as compared to the scrambled sequence, we had an increase in TREM2 positive cells, an increase in CD11. So we have more gliosis, actually, we activated cells. And these cells were actually better phagocytes. So we regained a part of a phagocytotic capacity in these cells. And what was really interesting as output is that we decrease the number of misfolded protein as well. So one of the disease-causing protein is misfolded SOD, basically, that it's causing the problem. So we had the antibody that was specifically recognizing misfolded SOD, and we see the basically decrease in the concentration of misfolded protein in the spinal cord. So what is our working model? A model at this time is that instead of attenuating immune response, what we want to re-establish functional immunity in the cells. So basically, once you have a chronically activated microglia, we have a basically increase of SRSF3 and increase of SRSF3 phosphorylation in these cells, which in terms will block translation and arrest translation of the transcript and blunt the TLR2 response here. And this leads to loss of phagocytic capacity and immune function, which in turn will exacerbate disease progression and survival. Once we target and silence SRSF3, we can functionally rescue, partially, not completely, but at least partially rescue microglial cells with diminished level of expression of SRSF3 and phospho-SRSF3. We can induce de novo synthesis of the targeted protein, and as by Western blot we can see, we can increase the TLR2 expression, rescue partially phagocytosis, and we can see that we can slow disease progression by increasing functional immunity in these cells. So this is our working model. So thank you for the, because this was kind of completely, we remade the slide within 5 minutes before the talk. And at this point, so what to sum up and what are the future perspectives. So what we can see here is that inflammation is really complex therapeutic target, and that the field evolves. So we are not talking now about eliminating inflammatory response. We can rather target dysregulation versus classical and conventional anti-inflammatory approaches, especially in chronic neurodegenerative disorders. So because we need that adequate and functional innate immune response is needed to kind of limit injury and disease progression in chronic neurodegenerative disorders. So we found that SRSF3 is one of the proteins that may act as a regulator, that acts as a regulator of innate immune genes, and there is a potential to use this as a new target, a new approach by targeting mRNA and translation to restore the proteome of the functional microglial cells. And what we are doing now currently, we are examining the role of SRSF3 in other brain pathology and to see whether similar pathogenic mechanisms and regulation of innate immune response are shares between different pathology and in aging. And here I would just like to thank the collaborators, to the people in the lab, and to the funding agencies. Thank you.